Hi everyone, this is a video presentation on Introduction to Transcription in general. So as we know that transcription is a universal event in all living organisms, from prokaryotic like bacteria to archaebacteria to the highest level eukaryotic organisms including plants and animals. So for the reaction, we need to understand certain basic factors which are closely associated with this reaction. So in this video presentation, we will be discussing three most important elements associated with transcription process. So here we can see that some of the basic features of the transcription will be described in this presentation, and also we'll have a discussion on the RNA polymerase enzyme, and finally about the promoter sequences. So before moving into the content, we know that central dogma is a common term used in molecular biology to discuss the feature of gene expression. So gene expression is what we refer the expression of a character in a cell and ultimately in an organism. So the gene expression consists of two important segments or two important phases. One is the transcription and the other one is the translation. So in the transcription process, we know that from a gene which is actually located in the chromosome of every organism. So from the chromosome, the genes which is actually a specified location, specified sequence within the bacterial chromosome or in any chromosome will be transcribed into a molecule called RNA. And that process is called a transcription, and from the RNA, the next level of the reaction in the central dogma is what we call translation. There, the informations which are stored in the mRNA will be converted into a polypeptide, and usually in the form of amino acids. So these polypeptide will execute a function, and that function is an attribute of that cell, or usually we call them as the phenotype of that organism. And conventionally we use the term genotype to represent the genetic sequence or the gene itself. So these are some similarities of transcription reaction seen in different forms of life. The most important thing is DNA is the template molecule in all organisms like prokaryote or eukaryote, and RNA is the product, and also the composition of RNA is the same, and the enzyme associated with the reaction is in all forms it's called RNA polymerase enzyme, even though there will be slight differences between them. So even we can see certain similarities, there are differences also. So what are the major differences between, basically we'll be focusing the differences between the prokaryote and eukaryote. So the most important difference comes in the site of transcription. So the site of transcription is not the same in prokaryotes and eukaryotes. In bacteria, the site of transcription will be the cytoplasm. As all of us know that there is no nucleus or any membrane bound structures inside. But in case of a eukaryotic organism, there is a clear nucleus, and hence the chromosomes are also located inside. So definitely the transcription is also located or occurring inside the nuclear plasm, and after that the mature RNA molecule will be transported into the cytoplasm for further processing. And the second difference is in the coupling of the reaction with the translation. In bacteria, the transcription is coupled with the translation. That means that two reactions occur simultaneously, unlike in eukaryotic system. And the types of RNA polymerase used are also different. In eukaryote, multiple RNA polymerase are being applied for synthesis of different types of RNA. At the same time, in Prokaryotes, only a single RNA polymerase is in function. The transcription factors involved are different. In bacteria, normally there is a transcription factor called the sigma factor, whereas in eukaryotic system we have multiple transcription factors, and each set of these transcription factors will be involved or will be associated with specific polymerase enzymes, and they are collectively called as general transcription factors. Then in the mechanism of transcription also, these two types of organisms differ. That means in the initiation and termination process they shows differences. In the regulatory mechanisms you can see difference. In prokaryotes you can see certain common mechanisms like operons. At the same time in eukaryotes usually hormones and other secondary molecules will control the mechanism. The primary transcript is different. In prokaryotic organism, the primary transcript is comparatively simple and is not supposed to undergo any kind of processing. But at the same time, in eukaryotic system, the mRNA is not a complete molecule and which has to undergo certain processing. Right, we will see what are the differences later. And finally, the post-transcriptional process, which are extensively found in eukaryotic system, not commonly found in the bacteria. Right, here you can see the difference between the mRNA of prokaryote and eukaryote. You can clearly see that in a prokaryotic system, the mRNA carries information for multiple proteins. You can see there is an alpha protein, beta protein and a gamma protein. So this mRNA molecule carrying information for multiple proteins are conventionally called as polycistronic. At the same time, a eukaryotic mRNA is monocistronic. And this is the coupling of the reaction. The transcription occurs at the top of the image. You can see the transcription machinery and the mRNA is being synthesized, and these mRNA will be occupied by the ribosome system, and that will be synthesizing the polypeptide, and this occurs simultaneously. This is what we call the coupling of the reaction with translation. And this image shows the same mechanism in eukaryotic system. You can see there is DNA molecule inside the nucleus and the transcription occurs inside the nucleus, and after the processing in the nucleus, the mature RNA molecule releases into the cytoplasm where they undergo the translation event. And now we have another comparison of transcription with a similar reaction called replication. So in both replication and transcription we are using a polymerase enzyme, but the enzyme used here is slightly different. It is an RNA polymerase and there it is called a DNA polymerase. And but the similarity is that both the enzymes will do a same reaction. That means both of them will catalyze the addition of a nucleotide into a 3' OH. The second similarity or the second comparison comes in the transcription factors or the proteins associated. In transcription, the proteins involved are called the transcription factors, whereas in replication it's called replisome. And here comes the third difference, or the third factor is very important, that RNA polymerase do not require any primer molecule for the synthesis to begin. That's an important difference. And in terms of template, both require template. Then there is single strand synthesis. In transcription, there is a DNA RNA hybrid formation in transcription. Usually short stretches of genes are being synthesized during transcription, unlike the very elongated molecule of DNA in replication. And finally, another important difference is that in case of replication, the process occurs only once in every cell, never twice or thrice. There is no multiple reaction for the replication, but transcription occurs multiple times even at a single location. That means whenever that gene need to be expressed, there will be gene expression. So these are the common comparison between transcription and replication. So let's move to the content. The first thing we'll be discussing will be the DNA dependent RNA polymerase. So this polymerase enzyme, it was discovered in 1960s. In the beginning of 1960s, it was first isolated from bacteria, followed by eukaryotes and archaebacteria. And the ultra structures are also available from different organisms, and number of copies are also pretty high in prokaryotic system. And the catalysis is the same as that of DNA polymerase, that there is an addition of a nucleotide triphosphate to a growing end of a nucleotide chain. So the reaction, we'll see that later. So the polymerase enzymes are present in all forms, but as I already noted, in bacteria we have a single type, but in eukaryotic system we have 3 different types. Each is specific for each set of RNA molecule. The RNA polymerase 1 is being dedicated for the synthesis of ribosomal RNA molecules, and RNA pole 2 for the mRNA, and pole 3 for the tRNA and small rRNA molecule like 5S rRNA. So these are the different types of RNA polymerase in eukaryotic system. And here comes the reaction which is going to catalyzed by this enzyme. You can see there there is an addition of a nucleotide into the 3' OH group of an existing nucleotide. And when you see the structural features, it's a heavy molecule in terms of molecular weight and also in terms of their subunit number. It's having somewhere around 460 kilodalton with 6 different subunits, and we can split them into core enzyme and the sigma factor when it comes as a hollow enzyme. So a hollow enzyme is formed by combining the core enzyme with the sigma factor. So sigma factor is an important element going to control the reaction of transcription. So this shows us a comparison between the different subunits of the polymerase enzyme in bacteria, archaebacteria and eukaryotic system. So the core subunits are almost present in all organisms. That means the homologs of these subunits like beta dash, beta, alpha 1, alpha 2, omega, all these subunits are present. That means homologous subunits are present in different systems. Like archaebacteria, you can see the similar set. Then RNA pole 1, 2, 3. In all sets, we can see the different homologous subunits for this bacterial subunits. So studying any one set is relevant to understand all other types of polymerase enzyme. This is the comparative image. I am not focusing on them. And when you see the subunits in specific, the alpha subunits are required for the assembly, the initial assembly of the enzyme. And moreover, the alpha has got the carboxy terminal domain and the amino terminal domain, and of which these subunits or these carboxy terminal domain are important for the interaction with the promoter site, right? And the beta and beta dash, which are actually the largest subunits in the polymerase enzyme, they function as pincer molecules and helps in opening the duplex. Omega is also an accessory kind of molecule supporting the assembly of the enzyme. And finally the sigma, which directs the enzyme to occupy properly in the promoter site, and they interact usually with the promoter region with some molecular interaction. So this is the pattern how this enzyme undergoes its assembly. The alpha subunit comes first, then followed by beta, beta dash and omega. And when you see in detail about sigma factor, sigma is the major transcription factor in prokaryotic system, and this one is primarily involved in recognizing the promoter sites of a gene. So every gene we'll come to the discussion of promoter later. So for every gene we have a promoter region. So these promoters will be identified or recognized by the sigma factors. So the sigma factor is important to recognize the promoter and followed by the gene expression. So without recognizing the promoter, no gene expression will be done in a system. So it's important. So the sigma factor which are present at all the time, or as a housekeeping sigma factor present in bacterial system, is what we call sigma 70, and the 70 represents the molecular weight. And based on that, again, we have different types of sigma factors like 70, sigma 32, sigma 28, sigma 54, etc., etc. So each of these sigma factors are associated with different functions also. So about the promoter region, we know that when you see the structure of a gene will have a coding region. So apart from a coding region, we also have a promoter region and a terminator site. So the promoter region is important for the beginning of transcription, and terminator region is required for the termination of the sequences of the transcription. So these two regions, even though it's not the intact part of the coding region, these are being counted as part of a gene. So conventionally we have certain notations also used in the gene. That means the region where it is undergoing the transcription site, or the point from where the coding region comes, is usually denoted as the +1 site. So the +1 site means from that particular base the transcription is going to initiate, right? And those sequences after that +1 site are given a notation of plus signal, plus symbol, and also it is called downstream sequences. So all the downstream sequences will be using a plus symbol. That means +2, +5, +10, etc., etc. Similarly, the other end of the sequences are called the upstream sequences, and they given a notation of a minus symbol. So there will be -10, -20, -30, -35, etc. sequences before the +1 site. Again, we can also see another important thing, that out of the two template strands, only one of the strand will act as the out of the two DNA strands, only one will act as the template, and those sequences which act as the template is also called as the non-coding strand, and the opposite strand of the template is called non-template or the coding strand. Why it's called coding? Because the mRNA synthesized will have the same code as that of a non-template strand, except that there will be uracil in place of thymine. So when you see in detail about the promoter of bacterial system, you can see two important regions. One is at the -10 position, the other one is at -35 position. So most of the promoter sequences are located in the upstream site, right? So one is at -10 position, the other one is at -35 position. So we have a closer look here. So the sequence at -10 is actually, it is the sequence like TATAAT. It's a consensus sequence, and that particular sequence is otherwise also called as Pribno box, right? And at the same time, at -35 region, we have another consensus sequence called TTGACA. Here again another look for the bacterial promoter. You can see the +1 site, also the -10, -35, and also another additional element is also given there called up element, which is actually an additional DNA element that increases the polymerase binding by providing the additional interaction site for the RNA polymerase. Actually, the alpha subunit of the RNA polymerase interacts with the up element, and the sigma factor interact usually with the -10 and -35 regions. And we are moving into the promoter sequences in eukaryotic system. Even though we know that there are different polymerases in eukaryotes like pol 1, 2 and 3. So for each polymerase enzyme, different promoter also exist. But here we will be seeing the structure of eukaryotic polymerase 2 core promoter region. So we have, this is a detailed promoter. That means we have 4 or 5 different set of promoter sequences. The +1 site is otherwise also called as initiator sequence, and there is a data box which location is at -25, and there is a BRE element above -30 sequence, and we have a downstream promoter element and also an MTE motif 10 elements. All these are different sequences which are located in the eukaryotic core promoter or the eukaryotic promoter for RNA polymerase 2. So this is again will be showing two sets of or two types of two extreme levels of promoters in eukaryotic system. That means a simplest form of promoter in which only a tata box is or a tata sequence is given, but in the second one there is a multiple sets of promoters are available. So this is about the introduction to transcription. So what we have seen in brief is that we have the gene here, we have the DNA molecule. So from this in the DNA, suppose this is the gene, and within this gene we know that certain regions are denoted like see, suppose this region is the up element and this one is the -35 region and this one is the -10 region. So these are the two regions which are, and this one is the +1 site, suppose. So in this case what happens is the polymerase enzyme usually comes, the polymerase enzyme with its different subunits, like usually the sigma factor will bind over here, the sigma factor, right, and followed by the alpha subunit. Usually the alpha subunit bind over here. Then there will be the beta dash, which is the biggest subunit, beta dash. Then also the beta subunit, and also there will be the omega. So all these different subunits interact with the promoter region, and this causes the initiation to begin, and that leads to the initiation. Then there will be synthesis of RNA molecule. There will be synthesis of RNA molecule from the gene. So the sequences from this +1 site up to this region will be synthesized, right? And once it reaches this particular site, there will be termination because there are terminator sequences. So this is what we have seen in this particular presentation, and after that this undergoes translation to produce what? Protein. So you will get proteins after the translation event. So this is what we have seen in this video presentation, and we have seen in detail about the RNA polymerase enzyme and also about the promoter sequences. So by that we'll wind up this presentation session, and I hope you understood, and thank you for watching.